My name is Michael Fox. I'm here today to talk to you about moving from correlation to causal mapping of human brain function. I want to thank the organizers of OHBM for the invitation to give this talk today. As disclosures, I do have some intellectual property and my funding sources are listed. So a question that I think everyone in the Organization for Human Brain Mapping is interested in is the question of how do we map human brain function? Now, this is a question I was very interested in during my PhD, and it's a question I remain very interested in now as a neuroscientist. But there's a very related question that I'm even more interested in as a clinician, which is not just how do we map human brain function, but how do we localize neuropsychiatric symptoms? And this goes beyond being just a neuroscience question. This is something that's got clinical and therapeutic relevance. In other words, I don't want to just map a neuropsychiatric symptom or a human brain function because I'm interested in figuring out where it is. I want to map these symptoms because I want a therapeutic target. I want to identify a spot in the brain, let's say the spot that's responsible for Duff beer, because I want to be able to stick DBS electrodes in that spot and turn off excessive Duff beer interest. Or conversely, I want a spot in the brain where I can hold a transcranial magnetic stimulation coil and turn off excessive craving for donuts. So how we go about localizing neuropsychiatric symptoms is something that my lab is very focused on. And the most intuitive approach for this, and the way that's dominated the field of functional neuroimaging for a long time, is neuroimaging of symptomatic patients. It's the most natural thing in the world. If you want to localize a neuropsychiatric symptom, find some patients that are suffering from that symptom, put them in a scanner, and see what brain areas have activity that correlate with the symptom that you're interested in. And this has been a very valuable approach that's given us a lot of insight into the brain regions or brain networks that are involved in different symptoms. But there are some important limitations of this approach to symptom localization. And the first problem is reproducibility across different studies. When we try to localize a symptom and then we try to do it again, we don't always come up with the same answer. And I'll just use one example, and this is localizing the symptoms of Alzheimer's disease. In fact, Alzheimer's disease of all neuropsychiatric conditions has got one of the most reproducible set of neuroimaging findings there are. But even in Alzheimer's disease, if you do a study trying to localize neuroimaging abnormalities that might be associated with the symptoms of Alzheimer's disease, you can get different answers. So this was a study that compared 30 patients with Alzheimer's disease to 30 age-matched control subjects, looked at cortical thickness, and identified abnormalities in the hippocampus, a very nice neuroimaging correlate of memory loss and Alzheimer's disease. A second group then repeated the same study with a different set of Alzheimer's patients and a different set of control subjects. And this time they got a different answer. This time the significant abnormality was in the posterior insula. And a third group did this study. In fact, we had 26 different studies that we looked at of people that identified neuroimaging abnormalities in Alzheimer's disease. And in this study, they used PET scans, and they identified significant metabolic abnormalities that were in the lateral temporal cortex. Now, again, study after study after study, we got valuable information about neuroimaging abnormalities in Alzheimer's disease, but they weren't particularly reproducible across studies. And in fact, you can test for reproducibility across studies using a formal technique, such as an ALE meta-analysis. And then in the case of these 26 studies of Alzheimer's disease, there were no consistent findings. In other words, across these different independent studies, there were not specific brain regions that were identified more than you'd expect by chance. Now, there are multiple different approaches to solving what's been called a reproducibility crisis in the field of human brain mapping. One is to improve our statistical approaches so that we get fewer false negatives or false positives. Another is to increase the size of our cohorts. Rather than studying 30 patients versus 30 controls, we can study thousands of patients versus thousands of controls. And all these are very valuable ways to go about improving our reproducibility. Now, one of the things that Ryan Darby did is ask whether we're looking for things to reproduce in the wrong way. Rather than test whether these different studies map to the same brain region with something like coordinate-based ALE meta-analysis, we can actually ask whether or not these different studies are identifying neuroimaging abnormalities that map to the same brain network. So what Ryan Darby did is he turned to something called the human connectome, and almost everyone listening to this talk is going to be familiar with this. This is an amazing resource that's been built by the human brain mapping community over the last few decades. By scanning thousands of healthy, normal subjects, either using MRI scans sensitive to water diffusion or MRI scans sensitive to spontaneous fluctuations in blood oxygenation or BOLD activity, we've been able to construct approximations of an anatomical wiring diagram of the human brain based on diffusion tractography, or what's been particularly valuable for us is a functional wiring diagram based on correlations in this ongoing spontaneous activity. And so what Ryan Darby did is he took each one of these studies that reported significant abnormalities in Alzheimer's disease, and rather than ask if the individual coordinates reported in these studies reproduced, he took each one of these coordinates and used it as a seed region for a functional connectivity analysis. In other words, he turned each one of these coordinates into a brain network, or the network of regions functionally connected to these coordinates. Then rather than ask whether the coordinates themselves are reproducible across studies, he asked whether the network was reproducible across studies. And what he found is very, very high consistency. So in contrast to the negative finding that he saw when he looked at the coordinates alone, when he tested whether these coordinates all mapped to a single consistent brain network, he found out that they did. In fact, 100% of these coordinates were functionally connected to the default mode network, or the hippocampal network, which is exactly the brain network that we think is related to Alzheimer's disease and Alzheimer's symptoms. Now, we've used this coordinate network mapping approach a few times. After Ryan's initial study focused on Alzheimer's disease, Ramona Whale came for a visit to my lab and focused on Parkinson's dementia. And just like in Alzheimer's disease, the coordinates of neuroimaging abnormalities in Parkinson's dementia have been reported all over the brain in lots of different brain locations, fueling a lot of debate as to where Parkinson's dementia might localize to. And when Ramona ran these coordinates using this network mapping technique, what she found is that all of these heterogeneous results across all these different studies that appeared to be poorly reproducible actually all mapped very nicely based on a network connected to the hippocampus. Matt Burke and my group applied the same coordinate network mapping approach to migraine. Again, lots of different neuroimaging abnormalities have been reported in migraine. And when he looked at what each one of those coordinates was connected to, he identified consistencies in the posterior insula, which is very involved in pain, and actually negative correlations or anti-correlations to visual cortex, which for anybody who works with patients with migraine or studies migraine knows visual aura is a very classic symptom or part of migraine where you can see flashes of light prior to the migraine starting. And in fact, our brain stimulation therapy for migraine, where we hold a TMS coil up to the back of the brain and try to stimulate area V3A, is exactly where his coordinate network mapping approach pointed us to. Again, linking different neuroimaging abnormalities that have been seen in migraine to a common brain network centered on regions that we think are involved in the symptoms of migraine, such as pain and visual sensation. So I'm going to return to this question again. How do we localize neuropsychiatric symptoms? And again, we're talking about neuroimaging of symptomatic patients having a couple problems. And the first problem was reproducibility. And again, there are lots of different ways you can go about solving it. One way we've looked at is seeing if things reproduce to a brain network rather than an individual brain region. But no matter how large you increase your cohorts, or how much you improve your scanners, or how creative you get for how these things should reproduce, there's another problem with this approach. And that's, in the end, we're still looking at neuroimaging correlates of the symptoms we're interested in, not causes of the symptoms we're interested in. Now, again, this might not matter if what you want is a diagnostic biomarker. If you're trying to know or predict whether someone's going to get a particular disease, a neuroimaging correlate can be perfectly good and highly valuable. But if what you want is a treatment target where you can intervene to make symptoms better, then this issue of correlation versus causation becomes a critical limitation of functional neuroimaging. And so over time, we've actually, in my lab, shifted our focus from neuroimaging correlates of different brain diseases to focus on causal sources of information for human brain mapping. Causal ways that allow us to link focal brain abnormalities to symptoms in a causal way rather than just using correlation. And there's actually a very finite number of sources of information that we can use for causal mapping of human brain function. And I'm going to talk about brain lesions and brain stimulation. So by far our oldest source of mapping out human brain function in a causal way is brain lesions. And in fact, mapping out human brain function based on lesions predated functional neuroimaging by decades, if not by centuries. And this is how lesion-based localization of human brain function is supposed to work. You take 3 patients, patient 1, 2, and 3, that all have focal brain damage that led to a new onset neuropsychiatric symptom. In this case, let's just say that this is depression. So all 3 of these patients had no history of psychiatric illness. They all had a stroke, focal brain damage, that led to new onset depression. Well, you can map out the location of each one of these patients' focal brain damage, map that brain damage to a common atlas, and see where these lesion locations intersect. And that one spot in the brain where these 3 lesions that all cause the same symptom intersect gives you a very strong causal link between that common area of brain damage and the resulting symptom. Now, unfortunately, this is how lesion localization is supposed to work. This is how lesion localization often works in practice. You take 3 patients that all have focal brain damage leading to the same new symptom, you map out the location of damage, and the damage fails to intersect. And this holds true for depression, for memory, for Broca's aphasia, for almost any symptom you'd want to look at. If you actually map out the lesion locations causing the same symptom, they don't map to one consistent brain region. And so just like we did for trying to make sense of heterogeneous coordinates in different brain locations, we turn to the human connectome. And again, we're going to do the same thing for heterogeneous lesion locations causing the same symptom. And using this very valuable wiring diagram, now we can turn back to our problem of lesion localization. And for the first time, rather than ask whether lesions causing the same symptom map to one specific brain region, we can ask the question, do lesions causing the same symptom map to one connected brain network? And now that we have this wiring diagram of the human brain, we're poised to begin asking this question. And so this is a technique called lesion network mapping that was developed by a fellow in my lab named Aaron Bose. And this was actually the index case that led to this approach, that led to what's now a field. This was a patient that came in with acute onset visual hallucinations. No history of psychiatric problems. Was a young woman driving in her car when bang, all of a sudden she looked out her window and the entire countryside looked as if it had been drawn in by crayon. She said, okay, this can't possibly be right. Pulled her car over to the side of the road, ended up presenting to the neurology clinic at Mass General, got an MRI scan, and sure enough, she had a focal stroke in her medial thalamus. which is that little red area that you can see here. Now, what Aaron Bowes did was he said, okay, I realize that's where her stroke was, but that doesn't help me understand her symptoms. I want to see what that lesion location is connected to. Now, again, he can't answer that question in the patient herself because the location of the lesion is actually dead brain tissue. But what he can do is go to the normative human connectome, a database of thousands of healthy normal subjects, and study what this lesion location is connected to. So you can extract the spontaneous brain activity from the lesion location and ask what other areas in the brain are functionally connected to this location of brain damage. And you get areas that are positively connected, shown here in warm colors, and areas that are negatively correlated or anti-correlated to this lesion location, shown here in green and blue colors. And Aaron did this over and over again for 23 different lesions that he had found throughout the literature that all caused acute onset visual hallucinations. Again, these lesions were all in different locations, but when he mapped out the network connected to each lesion location and overlapped the networks, the patient's symptoms started to make a lot of sense. Specifically, 22 out of these 23 cases, the lesion location was negatively connected or anti-correlated to extrastriate visual cortex. What that means is that when brain activity at a lesion location goes down, and we think a lesion suppresses brain activity quite a bit, you'd expect brain activity in the extrastriate visual cortex to go up, just as a natural consequence of the way the human brain is wired. And riding on the success of Aaron Bowes' technique, we've applied this to lots of different neuropsychiatric symptoms at this point. And we think that this is a general principle of how to understand lesion-induced symptoms that seems to be applicable almost independent of the neurological or psychiatric symptom you might be interested in. And I'm going to highlight just a couple of examples during my talk today. So focusing in on amnesia, Michael Ferguson and Chun Lim wanted to look at lesions causing acute onset memory impairment. We talked earlier about Alzheimer's disease and neuroimaging correlates of Alzheimer's disease. These are patients with stroke that acutely lost their memory, their ability to encode information. And many of these lesion locations occur in classic areas of the memory circuit, or what's been called the circuit of Papes. A patient with lesions in the bilateral hippocampus, similar to patient HM. Patients with lesions in the anterior nucleus of the thalamus, or the retrosplenial cortex, or the mammillary bodies. But there are also lots of cases of lesions that caused a very similar acute onset amnestic syndrome that didn't fall within this classic circuit of Papes or memory circuit. And so what Michael did is he took each one of these lesion locations, converted it into a network of regions connected to the lesion location, and looked for commonalities in the network. And he actually split his data set of 54 lesions causing acute onset amnesia into two groups, just to look at reproducibility. And what he found is that lesions causing acute onset amnesia were all connected to a very specific brain circuit centered on the subiculum, but also extending into the retrosplenial cortex and involving other brain areas. And when he zoomed in on the most sensitive and specific connection for lesion-induced amnesia, this was comparing his amnesia lesions to lots of other lesion locations that didn't cause acute onset amnesia, what he found is a critical hub at the subiculum retrosplenial junction. This is a single spot in the brain that was connected to every single lesion location that causes acute onset amnesia and wasn't connected to other lesion locations not associated with memory impairment. And so he can go to this critical hub in the subiculum, look at functional connectivity with this location, and that defines a brain network that by definition encompasses his 54 lesion locations causing acute onset amnesia. And you can see the intersection of the original locations here in white with his memory circuit defined based on connectivity of the subiculum in orange and yellow. He then went to two different independent data sets of patients that had focal brain damage and then had their memory tested, but didn't come in complaining of severe amnesia. He then asked whether intersection of these patients' lesion locations with his memory circuit would predict their memory score on a continuous basis. And this is just showing a few patients from these new data sets. These are the 5 patients that had the worst memory score in one of our data sets. And you can see all of their lesion locations intersected his memory circuit. And these are the 5 patients with the best memory score in one of these independent data sets. And you can see that those lesion locations all spared his memory circuit. In fact, across these two independent data sets, he was able to predict memory scores on a continuous basis just based on how much their lesion location intersected his memory circuit, derived from lesions causing a severe form of acute onset amnesia. He also looked at whether or not his circuit derived from brain lesions lines up with what we know about memory from functional neuroimaging. So if you put normal subjects in a task or in a scanner and have them do a episodic memory task, you identify lots of neuroimaging correlates of memory, shown here in these purple dots. And all these neuroimaging correlates seem to intersect Michael's lesion-based memory circuit very nicely. Similarly, he looked at neuroimaging correlates of Alzheimer's disease, a neurodegenerative disease of a memory impairment, very similar to those coordinates that Ryan Darby had looked at earlier. And again, those coordinates of memory impairment in Alzheimer's disease map to the same circuit. And then finally, he looked at brain stimulation sites that have been associated with memory enhancement. Again, this is a very active research field trying to see, is there any spot in the brain where we can modulate brain activity and potentially improve function of the memory circuit. Again, that remains an ongoing topic of investigation, but the preliminary data of different sites that were able to improve memory also map to this circuit, suggesting that mapping symptoms to a circuit using brain lesions might identify a circuit for therapeutic intervention. Moving from memory into depression, and I'll actually show 3 examples of lesion network mapping today. So focusing in on depression and using the same technique, Jaya Padmanabhan asked whether or not lesions associated with new onset depression map to a specific brain region or a specific brain circuit. And this was a very difficult one. She actually ended up putting together 5 independent data sets across 3 continents to find enough lesions that were either associated with new onset depression or control lesions that were associated with no increase in depression after the focal brain damage. The first thing she did is just look at the lesion locations themselves. This has been a very controversial topic in the literature as to whether or not there's any specific spot in the brain that, when damaged, predisposes you to depression. There's been a lot of interest in the left dorsolateral prefrontal cortex based on early reports suggesting that when the left dorsolateral prefrontal cortex is damaged, you're much more likely to get depression than when other areas are damaged. And in fact, it's that lesion data that motivated early trials of transcranial magnetic stimulation to the left dorsolateral prefrontal cortex to get depression better. But when Jaya looked at this very, very large data set of lesions associated with depression or lesions associated with no depression, there was not a single spot in the brain where lesion location could be significantly linked to depression. And this included a priori ROIs in the left dorsolateral prefrontal cortex. However, when she turned each lesion location into a lesion network and tested not whether there was a significant difference in lesion locations, but whether or not there was a significant difference in the connectivity with each lesion location, then she got a very robust result. And sure enough, that result was in the left dorsolateral prefrontal cortex. Now, again, what this means is that connectivity with the left dorsolateral prefrontal cortex defines a distributed brain circuit that best encompasses lesion locations associated with depression. So indeed, lesions to the dorsolateral prefrontal cortex are associated with depression, but it can be difficult to see that relationship statistically because lesions to certain areas of the parietal cortex or the temporal cortex or even the cerebellum are also associated with depression. And Jaya also looked at whether or not this brain circuit derived from lesions associated with depression was relevant as a therapeutic treatment for depression. And in fact, there's been 3 trials of transcranial magnetic stimulation to the left dorsolateral prefrontal cortex to try and help patients with post-lesion depression. And you can see the location of the 3 stimulation sites across these 3 trials here. All 3 of these trials used high frequency TMS designed to excite the left dorsolateral prefrontal cortex, and all 3 trials were successful. Now, the reason this example is important is it suggests that if we use lesions that cause a neuropsychiatric symptom to map out the brain circuit, that circuit might be therapeutically relevant to make those symptoms better. I'm going to show just one more example from lesion network mapping, but encourage people to look at all of our published papers if they're interested in a neuropsychiatric symptom that I'm not covering today. And that's tremor relief. And the reason I'm focusing on this example is that most of the times getting focal brain damage is a bad thing. Occasionally, focal brain damage can be a good thing. And these are all cases of patients that had severe essential tremor. So this is a disorder where when you reach for things or try to take a bite of food, you get very severe tremor of your hand. And then they had a stroke, and their functional use of their hand actually improved because these strokes all got rid of the essential tremor above and beyond any effect on hemiparesis or causing a paralyzed arm. And so these are all patients that actually did well following their stroke from the standpoint of improving the ability to use their hand. Now, these patients should tell us something really important about how we make essential tremor symptoms better in patients that have essential tremor. But if you look at the location of brain damage across all these cases, you're left scratching your head as to which one of these cases do we want to imitate if we're going to go ahead and create a surgical lesion to make tremor better. And so what we did is test whether or not these 11 cases of lesion-induced tremor relief map to a critical circuit, and maybe that circuit is our therapeutic target. And so that's just what Juho Jutsa did. And what he found is that all of the lesions associated with tremor improvement were connected to 2 very specific spots, one in the thalamus and one in the cerebellum. And actually, this location in the thalamus was very interesting to us. And the reason being is that spot in the thalamus overlapped exactly with the ventral intermediate nucleus of the thalamus, the VIN target that we use to implant our deep brain stimulation electrodes to provide tremor relief. And in fact, the peak of his lesion network mapping analysis was the same coordinate as the coordinate that we target our DBS electrodes to for tremor relief. Now, again, we already know where a good therapeutic target for tremor is. And so this is just telling us something or validating something that we already knew. But we identified a second target in the cerebellum that hasn't been tried for tremor improvement. It might be a second valuable therapeutic target in patients that don't get better from DBS to the thalamus alone. Further, this technique might be useful in other disorders where we don't know what the therapeutic target is. And so recently we've been looking at lesions that get rid of addiction to try and back out a therapeutic target for addiction using this same approach. At this point, I want to move on from brain lesions into brain stimulation. So I mentioned earlier that brain lesions were the original form of causal mapping of human brain function. But now we have a new approach that also gives us a causal link between a focal spot in the brain and a change in neuropsychiatric symptoms. And both deep brain stimulation and non-invasive focal forms of stimulation like transcranial magnetic stimulation highlight a spot in the brain that when modulated leads to symptom relief. So let's look in at brain stimulation for a minute, and we're going to focus first on deep brain stimulation. And as many of you know, deep brain stimulation is a highly effective treatment for the motor symptoms of Parkinson's disease. But just like neuroimaging coordinates and just like lesion locations, there's heterogeneity in the brain stimulation site. So these are all patients that were implanted with DBS electrodes targeting the subthalamic nucleus, which you can see there in red. And or actually, sorry, that's the red nucleus. The subthalamic nucleus is this white outline here that you can see in pale yellow. And you can see that even though surgeons are very, very skilled, not everybody ends up with their DBS electrode in exactly the same location. There's subtle variability in where that DBS site is. And what Andy Horn did is he looked at the variability in this DBS electrode position, and he calculated the location of stimulation in each patient, and then looked at what was connected to that stimulation site. Again, he turned a focal source of information into a network. He could then ask which network connections are associated with therapeutic response. And what he identified was a very specific set of functional connections with the DBS site that were associated with improvement in Parkinson's motor symptoms. And he also used a diffusion-based anatomical connectome to look at structural connections associated with improvement in Parkinson's symptoms. And actually, the functional connections and structural connections predicted independent variance in outcome. He then took these network maps of which connections you need to hit to improve Parkinson's symptoms and tested whether this connectivity pattern would predict outcome in an independent cohort. And actually it did, both for functional connectivity and structural connectivity. And these were DBS patients implanted at a totally separate DBS center that actually used a slightly different outcome measure. But again, DBS electrodes that were connected to the right set of brain regions or the right network were associated with better improvement in Parkinson's symptoms. You can use this same network mapping approach for transcranial magnetic stimulation sites. Now, the number one use of TMS isn't Parkinson's disease, but it's actually depression. But just like we had variability in coordinates, variability in lesion locations, variability in DBS sites, of course, there's variability in where each subject is stimulated when we treat patients with TMS to try and improve their depression. And this variability in the stimulation site seems to be related to the antidepressant effect of TMS. Now, just like we did for all the other modalities we've examined so far, we can use network mapping to see what network we need to be hitting to improve depression. And in this case, we had a very specific hypothesis of what brain region we thought should be critical for depression based on a wealth of functional neuroimaging data suggesting that almost every treatment that we use to make depression better, we see suppression of the subgenual cingulate. And a lot of this was Helen Mayberg's work and many others, really implicating this region in depression from a functional imaging standpoint. And in fact, this motivated trials of implantation of DBS electrodes in this subgenual cingulate location. And so we simply asked, does connectivity between each patient's stimulation site and the subgenual cingulate predict antidepressant response? And we found out in a prospective test of this hypothesis that the answer was yes. The more anti-correlated each patient's stimulation site was to this critical limbic region, the more their depression improved. And actually more exciting to us than our own results was actually a follow-up paper that came out very shortly after ours from a group down in Australia, Paul Fitzgerald's group, who conducted exactly the same trial and got exactly the same result. Again, suggesting that connectivity between the TMS site in the dorsolateral prefrontal cortex and this limbic region seems to be tightly linked to antidepressant response. However, you don't need to just focus on the subgenual cingulate. There was very good reason to focus on this region up front, but you can also do this in a data-driven manner. And that's what Sean Siddiqui and my group has been doing. So what Sean has done is he's taken each patient's stimulation site. You can see here there's 3 patients with heterogeneity in where they were stimulated, and compute the connectivity of each patient's stimulation site, and then just look for connections that correlate with antidepressant response. And you can see there that one of the important regions certainly is the subgenual cingulate, outlined here in red, but there are other important regions that come out. But one of the critical things that Sean discovered is that this data-driven map of connections that are important for antidepressant response seems to be different depending on what symptom of depression you're talking about. We know that depression is not a homogeneous entity. There are many different symptoms of depression that need not map to the same brain circuit. And in fact, this is a map of which connections are most important to improve the symptom of decreased interest. And when he looked at other symptoms, such as which connections are most important to improve feelings of failure, you actually get a slightly different circuit map out that has much less to do with the subgenual cingulate, but does implicate other areas. And when he looked at some symptoms like sleep problems, the connections you need to hit there were very different. And in fact, some of the connections were actually in the opposite direction of the connections that you want to hit to improve other symptoms of depression. So after mapping out each one of these individual depression symptoms, Sean noticed something important, which is if you drop the threshold and you look at these connections that you need to hit to improve sadness or anhedonia or decrease concentration, it more or less was the same set of connections. And those connections were very different than the connections that you needed to hit to improve interest in sex or sleep or indecisiveness. And he ran some formal clustering analyses on these different circuit targets for specific symptoms of depression. And he found that in the end, there's really just 2 main clusters or 2 circuits associated with improvement in 2 sets of depression symptoms. One that we call the dysphoric cluster, and another that we call the anxiosomatic cluster, just based on the type of symptoms that seem to improve when you stimulated a spot in the dorsolateral prefrontal cortex connected to each one of these circuits. Now you can then take these circuits and actually identify spots on the surface of the brain in the dorsolateral prefrontal cortex that are best connected to each one of these circuit targets. So what you see here on the left is this is the set of connections. These are the spots in the dorsolateral prefrontal cortex that best represent the connectivity to that dysphoric circuit. These are the spots on the dorsolateral prefrontal cortex that best represent the connectivity to the anxiosomatic circuit. And in fact, because these 2 sets of connections were very complementary, Sean actually just combined them into a single map, where if you want to improve dysphoric symptoms of depression, you're going to want to stimulate the red and yellow areas. If you want to improve the anxiosomatic symptoms of depression, you want to stimulate more the pink and purple areas. And in fact, he performed this exact same analysis in an independent data set and got exactly the same result, suggesting that this is something that is reproducible, even though all these analyses were retrospective. And in fact, he could use this map of dysphoric versus anxiosomatic symptom improvement, build the map based on one data set, and use it to predict which set of symptoms were getting better in the other data set. So returning back to our causal sources of information, we talked about the value of brain lesions and how the human connectome can be used to map out what network lesions causing the same symptom are hitting. We've talked about deep brain stimulation and transcranial magnetic stimulation, showing that the human connectome can be used for the same thing to map out connections associated with symptom improvement. But what if you put everything together? Will all of these causal sources of information actually converge on a single causal substrate for a symptom that we're interested in? And we've just begun to start to explore this question and put all the data together. And the first one we focused on, again, the work of Sean Siddiqui, was depression. And so at this point, we have 14 independent data sets. So 5 cohorts of brain lesions, 465 individual lesion locations, 4 cohorts of TMS data where they recorded the exact stimulation site, so 151 TMS stimulation sites, and 5 cohorts of DBS patients, where patients were implanted with DBS electrodes either to improve depression, such as these data sets with DBS to the subgenual cingulate, or actually, in some cases, DBS cohorts associated with DBS inducement of depression. Either Parkinson's patients where turning on the electrodes made depression worse, or in the case of epilepsy patients implanted in the anterior nucleus that made depression worse. And across these 14 data sets, we can back out connections associated with lesion-induced depression, TMS-induced depression improvement, or DBS-induced depression improvement, or depression exacerbation. And we can derive a causal depression circuit based on lesion data, TMS data, or DBS data. And what we found is that the 3 circuits derived using these 3 independent causal sources of information reproduced extremely well. In fact, the circuit derived from these different modalities was nearly identical. And so Sean has now put all of this data together and derived a single depression substrate based on these 14 independent data sets. And you can see that there are a lot of regions in here that we already thought were involved in depression based on neuroimaging correlates. Areas like the dorsolateral prefrontal cortex that we use as a TMS target for depression. Areas like the subgenual cingulate that we are using as a DBS target for depression. But it's a circuit, not just individual brain regions. And in fact, when we looked at our brain stimulation data sets, we could show that across both TMS data and DBS data, the more connected your stimulation site was to this circuit, the more modulation of depression we observed. And in fact, connectivity to our circuit worked better than connectivity to any individual brain region. So I want to leave you with 3 conclusions, and I hope I motivated some interest and some questions. Conclusion number 1 is that the human connectome is useful for linking neuroimaging coordinates, lesion locations, and brain stimulation sites to brain networks. Conclusion number 2 is that lesions and brain stimulation sites provide causal links to human neuroanatomy. I think this is a uniquely valuable source of information. And take-home point number 3 is that causal network mapping of human brain function may help us identify therapeutic targets for neuropsychiatric symptoms. And we're now very focused on translating these network maps that we've identified into therapeutic trials. I want to acknowledge a huge amount of work. I'm privileged to work with some of the most brilliant students in the world. Every day I get to sit in my office, have people knock on my door and come in with another great idea or great result. Both my current lab members as well as former lab members that have moved on and are actually running independent research enterprises of their own. I also want to acknowledge all of my collaborators. As you saw, I presented a huge amount of data, lesion data, DBS data, TMS data. My lab would get nowhere without data sharing. And this is just a partial list of people that have been very generous about sharing data and working with my lab to combine data sets and try to localize neuropsychiatric symptoms. I also want to thank the neuroimaging community and the Organization for Human Brain Mapping as a whole. Initiatives like the Human Connectome Project or the Brain Genomics Superstruct Project from Randy Buckner, and that's the connectome data set that I use for most of my work, have given us maps of human brain connectivity. They've given us a wiring diagram of the human brain that's helping clinicians like me begin to sort out how we make our network targeted brain circuit therapies even better. At this point, I'd like to take any questions. I believe that there is a chat room that will be opening up. I want to solicit any postdocs or collaborators that would be interested in working with me. I'm always looking for talented new people, and we'll shortly be starting a new center centered around brain circuit therapeutics. And please reach out to me if you'd be interested in working with me or interested in a collaboration with these types of approaches. And thank you again for having me speak today, and I hope that there are some questions.